Good afternoon, all.
Good afternoon, sir.
Good afternoon, sir.
Am I audible?
Yes, sir.
So, what is the last class in plant physiology? Yesterday, what Nagaraj sir did tell? What explained?
Photosynthesis.
Photosynthesis completed. Photosynthesis entire photosynthesis topic completed or not? Didn't attend yesterday class?
No, sir.
Why didn't attend most of the students in this online classes? Only this is only 10 to only the purpose those who are not unable to attend the online class offline classes. That's why we tend to establish this online classes. Why didn't attend?
Yesterday is working day, sir. Second Saturday, though also government give you permission to the schools for working day, sir.
Today also I have seen in C1 where it only 10 or 12 members. I don't know C2 and C3. How many of you attended? Please utilize this opportunity.
Yes, sir.
Yes.
How many of you attended offline classes?
Me, sir.
So offline in plant physiology, what topics are completed?
Light reaction, dark reaction, completed.
In offline,
Offline.
are you able to see this slide?
Yes, sir.
Yes, sir.
So in plant physiology topic, already clearly explained in offline classes. Those who attend offline, they know the concept of plant physiology and steps in plant physiology, how to read, how to prepare plant physiology. My voice is clear, first of all,
Clear, sir.
right? So plant physiology syllabus organized well. The first plant water relationship, already it was completed offline classes. And next photosynthesis, and next respiration,
Respiration.
and nitrogen metabolism, and plant growth, promote regulators, means promoters and inhibitors. So these are the main aspect in plant physiology. Physiology means physiological activities, metabolic activities. How metabolic activities happen inside the plant body. So for any metabolic activity, here the required water. That's why the water importance and water relations placed in the first block. The entire water plant relationship based on the water potential properties. Already know what is water potential properties, and also how the water absorb, how the water movement, and the water move upright direction from root to the leaf, how they are evaporate. These are all topics completed.
Yes, sir.
And also yesterday Nagaraj sir also clearly explained about photosynthesis. So for plant growth, water, minerals required. Apart from the water and minerals, and also food material also required. Not only plant, any organism required the nutrition. Likewise, plants also required nutrition in the form of carbohydrates. But how they get the carbohydrates? How the plants get carbohydrates?
Process.
Can they depend on other animals?
They prepared by themselves.
They can prepare their own food material as they autotrophic nature.
They make their own.
As the autotrophic nature, they can prepare their own food material. That process called as photosynthesis, right?
Yes, sir.
So in photosynthesis, two stages, light reaction and dark reaction. Anyway, with help of these two stages, two reactions,
Yes.
carbohydrates prepared. So after formation carbohydrates, what happened here? This based on the carbohydrates, actually here the carbohydrates or proteins or fats act as a substrate for the respiration. So in this topic in respiration, actually carried out by three stages. One is glycolysis, another one is Krebs, and electron transport system. Let me give to some importance here. So first of all in plant body, with help of the sun, what happened here? ATP and NADPH assimilatory power formed in this is the first reaction, photosynthesis. Utilize this ATP NADPH as a result formation of glucose C6 H12 O6. First of all, where this happened, this reaction happened in this light reaction happened, and this formation of food material happened inside the where this all happened. Hello?
In thylakoid.
Right, in thylakoid. Thylakoid is part of
Chloroplast.
right chloroplast. That means, for example, let me change color. So this blue color border, this complete, for example, chloroplast structure. In chloroplast structure, for example, this formation and light reaction mainly happened in, as you said, happened in thylakoid membrane. Like this thylakoid membrane. Except thylakoid membrane, remaining the empty place called as stroma. This empty place called as stroma. Now, after light reaction in stroma, ATP, NADPH assimilatory power formed. And this ATP, NADPH utilized for the formation of glucose. Now, where is glucose in chloroplast, right?
Yes, sir.
Where is glucose? Glucose in chloroplast. Now, after formation, this glucose utilized plant body for respiration and also excess of the food material storage in the plant parts. So first of all, after formation the glucose, now this glucose act as a substrate for the respiration. Immediately inside the chloroplast or this glucose diffused into cytosol. Cytosol means, so this is the blue color chloroplast. Let me take another color. Here the pink color, for example, this pink color portion is mitochondria. Where is respiration takes place? Where the respiration takes place? Hello?
Mitochondria, sir.
Right, mitochondria takes place. Before mitochondria, what happened? First, after the diffused into cytosol, this between the place, the chloroplast and mitochondria, in between the place, this is called cytosol. In cytosol, after reach the glycolysis, what happened? Sorry, after reach the food material, carbohydrates, first stage of respiration takes place. The first stage of respiration called as glycolysis. The first stage of respiration called as glycolysis. What happened in this glycolysis first? And here the one another important thing. The same happened in animal cell also, but the one difference is the glycolysis may happen in cytosol or in chloroplast in plant cell. In plant cell, glycolysis takes place. That means initiated in chloroplast itself, or initiated in cytosol also. But in animal cell, always the glycolysis takes place inside the cytosol. Then what happened first? Let's see in glycolysis. In the name given itself, glycolysis. Glycolysis means breakdown
Breakdown.
of the glucose, carbohydrate, glucose into two molecules of pyruvic acid. That is the end product. Two molecules. How many molecules? Two molecules of pyruvic acid with its 3 carbon product. With its 3 carbon product under aerobic condition. And here the respiration two types, aerobic and anaerobic. I will explain later anaerobic respiration. So in this glycolysis reaction, the common in aerobic and also anaerobic. Aerobic and anaerobic. But in aerobic, another difference, after formation glycolysis, next Krebs cycle takes place, but the Krebs cycle absent in anaerobic because lack of oxygen. Krebs cycle. Based on the Krebs cycle, electron transport system or oxidative phosphorylation happen. So finally, what happened? The ultimate product is ATP energy produced. ATP energy produced. So through respiration, what happened? First, ATP energy produced. How ATP energy produced? By initial reaction of glycolysis and also Krebs cycle reaction, and finally oxidative phosphorylation. Due to oxidative phosphorylation, ATP energy formed. Finally, ATP energy formed. So this is our concept. Already first we see the concept of photosynthesis. Light, with help of light, ATP and NADPH formed through jet scheme. From the ATP and NADPH, glucose formed. Now this glucose act as a substrate of the glycolysis or respiration initial first step.
Yes.
So what happened in this? First, incompletely oxidized. That means is formed as a 2 molecule of pyruvic acid. From the 2 molecules of pyruvic acid, acetyl coenzyme is formed. This is connecting link between glycolysis and Krebs. I will explain later. From the Krebs, an NADH, FADH formed. These are entering into the electron transport system and finally form the ATP. So I will explain one by one clearly. First of all, the first step of respiration, that is, what is that? First step of glycolysis. First stage of respiration, glycolysis. First stage of respiration, glycolysis. Let's start with glycolysis, right? So as we discussed earlier, glycolysis breakdown into 2 molecules of pyruvic acid, simple. So this pathway described by Emden Mayer of Parnas. Emden Mayer of Parnas, and it is also called as Emden Mayer pathway, EMP pathway, another name. So this is called as EMP pathway. In examination, if they ask EMP pathway, glycolysis or respiration first stage, any any type of, the way of question different, but answer is one. So in examination may ask about direct question glycolysis or describe respiration initial stage or initial process, initial steps or EMP pathway. So this is about the glycolysis introduction. Let's see the steps. Glycolysis steps. Glycolysis steps. So first, as we discussed, glucose is the product. Not only glucose, may also be proteins and fats also. Right. Right. Are able to see this?
Yes, sir.
1 minute. Right. Are able to see this glycolysis steps? Hello?
Yes, sir.
Yes, sir.
Yes, sir.
Right. As we discussed earlier, this is the substrate. Already you know the formula C6H12O6. In examination, if possible, draw this chemical structure. If not possible, you can draw like this. Actually, this is not clear. You can draw C6SC2, 3, 4, 5, 6. C6, H12. You can write this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Like this H12O6. Oxygen number also 6. Or you can just write simply C6, H12, H12O6. So this is the glucose, first substrate. First reaction is phosphorylation. What is the reaction name? Phosphorylation. Why it is called phosphorylation means, with the help of exokinase enzyme, from adenosine triphosphate, ADP, convert into ADP. One phosphorus. Actually here, how many phosphate? ATP means 3 phosphate. 1, 2, 3. In this 3 phosphate, one of the phosphate attached to the 6th carbon. Here you can see 6. This is 1, carbon 1, 2, 3, 4, 5, 6th. So this is 6th carbon. In 6th carbon, what happened? One phosphate group added. Here, this is the phosphate group. One phosphate group added at the 6th carbon. That's why it's called as a glucose common as it is. But what added in 6th carbon? Phosphate group. What added in 6th carbon? Phosphate group. That's why it is called as a glucose 6-phosphate. Right? So this is the first reaction. So this is the first reaction. What is the first reaction name? Write down. First reaction is phosphorylation. Remember it. First reaction name. In examination, you just draw this cycle steps and also elaborated with headings. What are the headings? First heading, phosphorylation. What happened in phosphorylation? Glucose convert into glucose 6-phosphate. That's it. Glucose convert into glucose 6-phosphate. Which enzyme involved? Exokinase. Exo means 6. 6. How many carbon number here? 6. So exokinase enzyme. What is the reaction of exokinase? Exokinase from the triphosphate, one of the phosphate transferred to the 6th carbon of glucose. When it transported to the 6th carbon of glucose, automatically it is turns into glucose 6-phosphate, right? So this is the first step. Hello, are you follow?
Sir, please
sir.
repeat it once again, sir.
Substrate clear first glucose C6H12O6.
Yes, sir.
Yes, sir.
Right. So your ATP A adenosine tri means how many phosphate tri means?
3
sir.
phosphate.
3 phosphate. 1, 2, 3. 3 phosphate are there. But what happened with help of hexokinase enzyme? This hexokinase enzyme transfer a phosphate of ATP toward the sixth carbon of glucose. At the sixth position phosphate group added, which means here in ATP one phosphate removed. So
అవన్నీ
just
ప్లేట్
assume
Yes.
డస్ట్బిన్లో వేసి ప్లేట్ నీట్గా కడిగి ఆడ పెట్టండి. కడిగి ఆనే బోర్లియండి మళ్ళీ.
please be silent. So anyway, in this triphosphate, one phosphate transferred. As a result, formation of this tri means 3.
Yes.
Later it's converted into
ADP.
ADP means
Yes.
Diphosphate. Adenine.
diphosphate. Right, diphosphate. Tri become di.
Yes.
Where is this phosphate? This phosphate transferred to the sixth carbon. That's why called as a glucose 6 phosphate.
Phosphate.
Is it clear?
Yes, sir.
This is called phosphorylation, first one. And second step, this is the second step. In the second step of glycolysis is isomerization. Then here the enzyme also involved isomerase. Isomerization. In isomerization, glucose 6 phosphate convert into fructose 6 phosphate. Which means actually glucose 6 phosphate, fructose 6 phosphate formula same. But what happened in isomerization? If you clearly observe here, so where is the keto group here? This is the first carbon. In glucose, this keto group means C double bond O. The C double bond O at the first carbon. C double bond O at the first carbon. But where is in fructose? The C double bond O?
Second.
Second carbon. So that changes happen, but never change the formula. The glucose 6 phosphate formula same C6H12O6. Fructose also same C6H12O6. But here the small changes are happened like this. So this is called as isomerization. The rest of the formulas as it is common. If you know the glucose 6 phosphate formula, the same fructose 6 phosphate formula. But one different is the keto group C double bond O first group shift to the second carbon. That's it. That is the different. Clear? So this is second. And third reaction again phosphorylation. Means already you know. Always whenever phosphorylation enzyme is same. What is the enzyme in phosphorylation? What enzyme involved here? Phosphorylation. What is the enzyme here? You see first reaction.
Hexokinase.
Right, here also hexokinase, of course same, but phosphofructose, because the substrate is fructose. Substrate name fructose. That's why we can mention as a phosphofructokinase. But mechanism same. What is the same mechanism here? ATP means
Adenosine triphosphate.
how many phosphate here?
Three phosphate.
3 phosphate.
Converted to 2 phosphate.
Right. This one on this 3, one of the phosphate add to the first carbon like this here. You can see in this image. Already sixth carbon. Let's change the color. Already sixth carbon have the phosphate from the beginning. The first phosphorylation where the phosphate attached at the sixth carbon. This is the sixth carbon. But in second phosphorylation in triphosphate, one of the phosphate transferred to the first carbon here. First carbon. Now how many phosphates are there in? How many phosphate groups? First carbon, sixth carbon, 2. That's why the same name Actually, this fructose 6-phosphate, the same fructose common, but where are the 2-phosphate group? First carbon. That's why we mentioned the first, and also sixth carbon. How many phosphate? 2. As it is consisting 2-phosphate, it is a name given as a bis or biphosphate. As a 2 biphosphate. If it is 3 triphosphate, if it is 1 monophosphate. So it is, as it is a 2, it is called bisphosphate. Bisphosphate. Right? Clear?
Clear.
Fructose bisphosphate. Now after this fructose bisphosphate, now aldolose reaction. Cleavage, that means later this converted as a cleavage. Next reaction is cleavage. So fourth one is cleavage. Fourth one is cleavage. You write down this all steps. So simple, fourth cleavage. What is the first step, first reaction? What is the first one?
Phosphorylation.
Very good, phosphorylation. What is the second one?
Isomerization.
Isomerization, good. And third one, again, phosphorylation. And fourth one, cleavage.
Cleavage.
So in cleavage, simply fructose 1,6 bisphosphate. Cleavage means what is the cleavage? Cleaved. Cleave mean break into two. Break into two, which mean here 3 carbon.
3 carbon compounds.
And here also 3 carbon compounds. Right? That mean fructose 1,6 bisphosphate, how many carbons here? How many carbons in glucose first of all?
6.
6.
6 carbon.
Glucose 6-phosphate also 6. Fructose 6-phosphate 6. Fructose 1,6 bisphosphate also 6 carbon. But due to the cleavage, this 6 become into
3 carbon compounds.
3 carbon and 3 carbon. Same the phosphate also split. That means 1, 2, 3. 1, 2, 3. First this 2, this let's change color here. Green color. 1, 2, 3. This 3 separated and remaining 3 also separated, right? 1, 2, 3, this carbon also. The above first 3 carbons called as DHAP, dihydroxyphosphate, dihydroxyacetone, dihydroxyphosphate. D means dihydroxyacetone phosphate. And another 3 carbon one called as glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. That is the fifth one. And again, sorry, that is the fourth one. And again, the fifth one here, isomerization takes place. Isomerization. Isomerization, I already told you what is isomerization.
Keto group shifted.
Right, that's it. So simple here, you can see here also. Glyceraldehyde, acetone phosphate, second carbon, the second carbon keto, right? Glyceraldehyde 3-phosphate, first carbon keto. That's it. And remember, this is the very, very important thing here. For further reaction, of course, here the 2 molecules are there, 3 carbon, 3 carbon. But the further reactions only carried out or further continued with help of the glyceraldehyde 3-phosphate only. But not DHAP. That means here the isomerization takes place, which means, so first of all, the glyceraldehyde 3-phosphate formed apart from the DHAP. First, this glyceraldehyde 3-phosphates undergoes to further reaction and formed as a one pyruvic acid. After what happened, this DHAP, dihydroxy acetone phosphate, isomerized into what? Isomerized into?
Glyceraldehyde 3-phosphate.
Right, very good. Glyceraldehyde 3-phosphate. Now this is isomer into glyceraldehyde 3-phosphate. When convert into glyceraldehyde 3-phosphate, the further process same. As these are all same dehydrogenase enzyme involved. Phosphorylation, enolose, same phosphate. So finally, what is the end product?
Pyruvic acid.
Pyruvic acid. Then how many pyruvic acid formed in glycolysis?
2, sir.
2.
That's it.
2
Two pyruvic acids are formed. 2 pyruvic acids are formed. They're very, very important. So only here the remember one thing, DHAP does not involve directly to formation pyruvate, because this, when the keto group second carbon is not involved in the reactions. When it is converted into first carbon, then only involved in further reactions. That's why DHAP isomerized into glyceraldehyde 3-phosphate. Let's see what happened after convert into glyceraldehyde 3-phosphate here. So the first NAD plus phosphate, cytosol phosphate, not ATP phosphate, and then some of the phosphate present dissolved in the cytosol also, cytosol phosphate added. As a result, formation of NADH plus H plus. NADH plus H plus. Simple. So actually, if you see in this first diagram, here the hydrogen is formed. Hydrogen is present. Glyceraldehyde 3-phosphate, hydrogen is present. But after converting to 1,3-bisphosphate, this is only 3-phosphoglycerate. 3-phosphoglycerate. 1,3-bisphosphate. 1,3-bisphosphate means, first of all, this hydrogen removed and this hydrogen added to the NAD plus. Now this NAD plus convert into NADH plus H plus. NADH2, NAD plus H plus. So now this hydrogen removed, right? Okay. In this next diagram, no hydrogen here. You can observe this. Apart from the what added here? What added here? Phosphate.
Phosphate.
Phosphate in the place of hydrogen. Hydrogen removed and added to the NAD plus and convert into NADH plus H plus. And in the same place, replaced by this phosphate. This phosphate. That's why already the third carbon have the phosphate here. Before split, this is the 6th carbon. Before split, before cleavage, this is the 6th carbon, right? Here.
Yes.
But after split, automatically after split, it is become 1 and 2 and 3. Already third carbon have the phosphate. But later, after remove the hydrogen, and this place replaced by the cytosol phosphate. That's why here, how many phosphate groups?
3.
Phosphoglycerate. In this phosphoglycerate, how many?
2.
Phosphate, 2 phosphate. One is yes. One is first carbon.
Other one is the
One is
3.
first carbon. Another one is third carbon. That's why 1,3-bis phosphoglycerate. 1,3-bis phosphoglycerate formed. Here the enzyme involved. Glyceraldehyde 3-phosphate is the substrate. Enzyme name dehydrogenase. Dehydrogenase. D means remove. D means remove. What remove here? What remove here? You can see here. What removed here? Hydrogen removed. Hydrogen removed. That's why this is called dehydrogenase enzyme. And this removed hydrogen added to the NAD and convert into NADH plus H plus. Okay? Right? After that, dephosphorylation. Dephosphorylation means what happened? This 1,3-bisphosphoglycerate, ADP, diphosphate, convert into ATP. Means di added by 1 phosphate, convert into triphosphate. Means ATP formed here. ATP formed here. So that means actually here the first carbon ATP, this first carbon ATP utilized by ADP. This ADP convert into di. After di means 2, after added 1, it is convert into tri. That's why if you see here the no phosphate, is there any phosphate in this 3-phosphoglycerate? Only 3-phosphoglycerate. That means only the third carbon phosphate is present. Only third carbon phosphate present, but not first carbon. Already first carbon dephosphorized. Dephosphorylation means removed from the substrate and added to the ATP. As it removed from the substrate, this is called substrate level phosphorylation. This is called substrate level phosphorylation or dephosphorylation. Substrate level phosphorylation or dephosphorylation. So anyway, here the formation 3-phosphoglycerate. 3-phosphoglycerate. If the 1,3-phosphoglycerate, 1,3 as it 2, it is called as a bisphosphate. Now 1 is removed. Only simply say as a 3-monophospoglycerate or 3-phosphoglycerate. Clear?
Yes.
And next one, mutation. Mutage. Enzyme mutage involved. Same in mutage. 3 carbons. Simple. The third carbon phosphate, you can see here. The third carbon phosphate mutation means changed. Changed toward the which carbon? You can see in this diagram. Third carbon shifted to the which carbon?
Second carbon.
Second carbon. That's it. Now that is called mutation. So here involved the mutase enzyme. Mutase enzyme. Phosphoglycerate mutase enzyme. Another one is dehydration. Dehydrolation means remove of water. Water removed. Enzyme is to remove the water. Enzyme is enolase. Enzyme is enolase. As a result, formation of phosphoenol pyruvate. Phosphoenol pyruvate. So after removal of H2O, you can see here. So this OH group is removed. H2O, this OH group removed. You can see, is there any OH group? Is there any OH group?
No.
Only H2O removed. Only H is present. So this is called phosphoenol pyruvate. Phosphoenol pyruvate. Now the last one again. So actually forgot one thing. The steps here. So how many stages so far completed? First phosphorylation, second isomerization, third again phosphorylation, fourth cleavage, fifth isomerization, and the sixth oxidation. Removal of hydrogen, oxidation. Removal of hydrogen called oxidation. After oxidation,
Substrate level.
this is S. Dephosphorylation or substrate level phosphorylation. Dephosphorylation or substrate level and mutation. Eighth is the mutation. Ninth, dehydration. And 10th again,
Substrate level.
very good. Substrate level phosphate. You can see in this diagram, same ADP diphosphate. ADP diphosphate converted into triphosphate. Where this phosphate added? From the second carbon. This phosphate, the second carbon phosphate removed here and added to the diphosphate, 2-phosphate. Now this diphosphate converted into triphosphate. You can see as a result formation of pyruvate. You can see, is there any phosphate group at second carbon?
No.
Is there any phosphate group? No phosphate group. Only one keto group is formed. This is called pyruvate. So how many steps involved in this glycolysis? 10 steps. Remember, 10 steps. In examination, if they ask about the glycolysis first step, EMP pathway, answer is, this is the answer. What is this? Once again, remember, glycolysis pathway, respiration first stage, or initial step, initial process, initial reaction, EMP pathway. So only steps. First, definition of glycolysis. Glucose split into
Breakdown. Glucose.
breakdown into two
Glucose.
pyruvates, two pyruvate acid. That is the definition. Or incompletely oxidation. Incompletely oxidation. That is also definition. So where it is takes place, cytosol. Who coined this stages, this cycle? Emden, Mayer of Parnas. So this is about glycolysis, right? Just simply, for example, first step, phosphorylation. You write glucose. No need to diagram this all chemical structure. If you interested, add it, no problem. Then, and this is also the remember of this chemical structure, not a big task. It is very easy. If you know one chemical structure, you can learn the another chemical structures also based on the one. Which mean where the phosphate added, where the phosphate removed, where the hydrogen added, where the hydrogen removed, where the water molecule removed, like this. So if you know one structure, you can draw. And previous 6 become 3. Simple. It is to make it off. If you see one structure, you can draw all the structures.
Yes.
If possible, better to draw in chemical reactions, chemical with chemical structure. Or else, if you know enough time, you just write phosphorylation, glucose convert into 6 plus, just write first reaction, enough. This is one reaction, this is one. And second and third, likewise 10 reactions enough with 10 headings. 10 reactions with 10 headings. But ultimate finally, pyruvates. Remember how many pyruvates are formed? 2 pyruvates are formed. 2 pyruvates are formed. My question is, how many ATPs are formed? How many ATPs are formed? How many ATP? In glycolysis, how many ATP are produced?
8.
How it is 8? Tell me.
3.
How it is 8? You can see in the image also. First, where are the ATPs formed?
4, sir.
First step.
First step, ATP formed or utilized?
Utilized.
ATP consumed or produced in first step? This step? Are you see
ATP breakdown.
Are you see this? Are you see this first ATP here?
Yes.
You tell me here the ATP produced or utilized?
Utilized.
Utilized, so not consider this. And where another ATP formed and this third reaction? The third reaction also ATP produced or utilized here? Utilized. And another ATPs are formed here. You can see here, this one. Here ATP produced or utilized? Produced, right? ATP produced. And another here last reaction. Here ATP produced or utilized? Produced. Now tell me how many ATPs?
2 ATP.
How many ATPs produced?
2
Think again. Think again.
4, sir. 4, sir.
How it is 4? How it is
times
4?
the cycle happens from cleavage
Right,
step.
very good. Very good. So already told you, one cycle from glyceraldehyde 3-phosphate, 2 ATPs formed. Another cycle after DHAP isomerase reaction glyceraldehyde 3-phosphate, again another 2. Total 4 ATPs are produced. But how many, out of 4 ATPs, how many ATPs are utilized?
2 ATPs.
Very good. 2 ATPs, that means minus 2. 2 ATPs are utilized. That what is the net ATPs, remember?
2 ATPs.
2 ATPs net. 2 ATPs net. But here another important thing is, so this is the direct ATPs 2, net ATPs 2. But actually this NADH2, are you able to see this marker? NADH plus H plus.
Yes, sir.
This NADH plus H plus oxidized as a result formation of a ATP. So one NADH2 oxidized, enter into the electron transport system. One ATP formed. One NADH2 oxidation. Sorry. 1 NADH2 enter into the oxidation, 2 ATPs are produced. I will explain clear in phosphorylation cycle. I will explain in detail in phosphorylation. 1 NADH2 involved in oxidation process, 2 ATP formed. So already direct ATP to clear net. Through NADH based ATP, how many NADH2 based ATP are produced? Huh?
3, sir. 1 NADH is equal to 3 ATP, sir.
No. That is in Krebs NADH2. I will explain clearly. I will explain later. That is in Krebs NADH2, 3. As you said, in Krebs NADH2 oxidation process, 3 ATP formed. But remember one thing, FADH2, glycolysis NADH2, only 2 ATPs are produced. I will clearly explain in ETS cycle. You don't worry. First remember, 1 NADH plus H plus oxidized produces 2 ATP. Likewise, how many ATPs are produced through NADH plus H plus?
4.
Very good. 4. How it is 4? Here only 1 NADH plus H plus.
2 times the cycle happens.
Yes, 2 times. Here also same 2 times. That's why first NADH plus H plus 2 ATP and second NADH plus H plus also 2 ATP. Total 2 plus 2, 4 ATPs are formed. Total 4 ATPs. This 4 ATP from NADH plus H plus oxidation process. And how many direct ATP? Direct ATP is 2.
4.
Now tell me total ATP member through glycolysis.
6.
6.
Right. Remember it. Remember the 6 ATP. 6 ATP through glycolysis cycle. 6 ATP through glycolysis cycle. Clear? Any
Yes,
doubt
sir.
in this? Hello?
No, sir. No, sir.
No, sir.
Clear,
No,
sir.
sir.
Right. Now next cycle. Of what is the next Krebs? Actually, before Krebs, one connecting link is there. I will explain later. First, is it clear? Pyruvate end product. Remember, end product is pyruvate. End product is pyruvate.
Pyruvate.
Okay.
Okay.
Right. Now, connecting link to glycolysis and Krebs. Connecting link glycolysis to Krebs. My voice is clear. Any disturbance?
No, sir.
No, sir.
Clear, sir.
Right. Already 2 classes completed. This is second class. First class complete. Only remain 2 classes. Please ask them to your friends, join the classes and utilize this opportunity. And what about the classes? All classes are good or any problem? Today morning, one of the posted the message, not understand well. I will add, and also I added the class also, and also I have checked up. You can ask directly. That is your duty. If it is not understand, to raise the doubts, you can raise the question. Clear?
Clear, sir.
Yes, sir.
Right. So what is the glycolysis end product?
Pyruvic acid.
Pyruvate.
Pyruvate, right? Pyruvate or pyruvic acid. How many? 2 pyruvate. 2 pyruvates.
Sir, voice is not audible, sir.
Not clear? Hello?
Yes, clear, sir. Clear.
So pyruvate. Now this connecting link, the pyruvate convert into acetyl coenzyme A. Acetyl coenzyme A. Pyruvate, how many carbons pyruvate?
3.
3
Yes,
carbon.
very good. 3 carbon pyruvate or pyruvic acid. 3 carbon pyruvate or pyruvic acid after oxidation.
Structure also in the exam.
In exam, that's what I told you. In examination, if possible, draw this structure, chemical structure,
Okay, sir.
or else you can directly write the name acetyl coenzyme A enough.
Okay, sir.
Cis aconitase, the isocitrate enough, citrate enough. That is your wish. That is a mandate. That is not a mandated to draw the chemical structure in here. So anyway, the end product of the glycolysis pyruvate. Now enter into the mitochondria. So far, these are all reactions taken in cytosol. Now cytosol to pyruvate enter into the mitochondria. What happened in mitochondria first? Decarboxylation. Decarboxylation means carbon dioxide removed. And also oxidation. And also
Oxygen is added.
oxidation process. Oxidation means NAD. NAD convert into NADH2. Convert into NADH2. Second, first decarboxylation. So decarboxylation, next oxidation. In oxidation from the pyruvate, hydrogen from the substrate, hydrogen removed, and this removed added to NAD, convert into NADH2. And also another step, another last step, add coenzyme A. Add coenzyme A. Coenzyme A. So this is the connecting link. So this after add coenzyme A, finally convert into acetyl coenzyme A. Now this pyruvic acid, 3 carbon pyruvic acid, convert into acetyl coenzyme A. Now tell me how many carbons are in acetyl coenzyme A?
2.
2. Why it is, how it is become 2 carbons?
From
Carboxylation.
Right, in the form of carbon dioxide removed 1 carbon.
Yes.
That's why 2 carbon compounds form. And also as it add coenzyme A, it is called as a coenzyme A, acetyl coenzyme A. Right? Now start the citric cycle. Krebs cycle. This is called as citric cycle because first formed citric acid, or another name called as Krebs cycle, or second stage of respiration. Respiration second cycle, second stage cycle. Respiration first stage cycle glycolysis. Respiration second stage cycle Krebs. Respiration connecting link, pyruvate decarboxylation and oxygenation.
Oxidative decarboxylation.
Yes, very good. So the name given decarboxylation means carbon dioxide removed. Oxidation means oxidation takes place. And also added coenzyme A. So these are all happened. So anyway, in Krebs cycle, first acetyl coenzyme A, 2 carbon compound, added to the 4 carbon oxaloacetate. 4 carbon oxaloacetate. This is the 4 carbon. You can see here the image also. How many carbons here? 1, 2, 3, 4. Right? The 4 carbon oxaloacetic acid or oxaloacetate condensed with 2 carbon acetyl coenzyme A. Already you know how the acetyl coenzyme A formed. How the acetyl coenzyme A formed? By the pyruvic acid. End product of glycolysis. Formed as the acetyl coenzyme A. Right? This 4 carbon oxaloacetic acid condensed with 2 carbon, as a result formation of 6 carbon. As a result formation of the 6 carbon citric acid. 6 carbon citric acid. 4 plus 2, 6 carbon. So here in this condensed process, Enzyme name is citrate synthase. Citrate synthase. Right? This is the first step. Condensed is the first step in condensed places synthetase enzyme involved. Dehydration or dehydrogenation here aconitase enzyme involved from the citrate water molecules as a result formation of cis aconitase formed as a cis aconitase. This second step. Third, hydration. That means add hydrogen, add water molecule. Here remove first reaction and second reaction again add. Here the enzyme name same for remove and added enzyme is same aconitase enzyme. In this time developed as a isocitric acid. Isocitric acid. Now tell me the carbon number in citrate. Cis aconitase. Isocitrase always sixth. Very good. 6 carbons. 6 carbon. So what is the fourth reaction? Oxidation. Here a fourth reaction is the oxidation. Fourth one, oxidation. 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 Oxidation means NAD. You can see NAD from the aconitase added hydrogen group to the NAD as a result formation of NADH plus H plus. NADH plus H plus. NAD after replace this from the substrate to hydrogen, here the remove hydrogen, that's why called oxidation. Remember it. Remove hydrogen oxidation. So the remove means dehydrogenase. Enzyme name dehydrogenase. Apart from the dehydrogenase, carbon dioxide also removed. Carbon dioxide removed. So as a result, so the complete reaction oxidation, not only oxidation and also decarboxylation. Oxidation and decarboxylation. As a result, formation of alpha ketoglutaric acid. Alpha ketoglutaric acid. Now tell me how many carbons in alpha ketoglutaric acid?
5 carbons.
Right, 5 carbons. 5 carbons because already 1 carbon lost in the form of carbon dioxide. Right, if you check this chemical structure also, COO common, CH2 common. Now this COO minus, only CH2 formed. Is there any COO group? CO2 group in this? No CO2 group. That was removed. The remaining as it is common. And also here the hydrogen also removed. You can see here the hydrogen in this here. In this diagram, hydrogen also removed. So this is the comparison of aconitase and alpha ketoglutarate. Now the fifth reaction, same oxidation. Fifth reaction also same oxidation. So what happened here also, NAD plus convert into NADH plus H plus. NADH plus H plus. Here also same hydrogen group loss. Hydrogen group loss. And add this hydrogen group. Add this hydrogen group to the NAD plus. As a result, formed as NADH plus H plus. Apart from that, here the same like a fourth one. Here also carbon dioxide removed. Carbon dioxide removed. As a result, formation of succinyl coenzyme A and added coenzyme A here and add coenzyme A. That's why it is called as succinyl coenzyme A. Then now tell me succinyl coenzyme A, how many carbons?
4 carbons.
4 carbons. Initial 5 carbon, but again remove the 1 carbon in the form of carbon dioxide. Again it becomes 4. 1, 2, 3, 4 carbons. And also here they added, you can see added coenzyme A. Added coenzyme A. So succinyl coenzyme A. That's the sixth one. From the succinyl coenzyme A, succinic acid form. This is the sixth one. This is the sixth one. And sixth reaction, like a phosphorylation. Phosphorylation means here the ADP in cytosol phosphate, not substrate. Here the not from the, is there any substrate in succinyl coenzyme A?
No.
Is there any phosphate group in succinyl coenzyme A?
No.
No phosphate, but the cytosol phosphate added diphosphate as a result formation as a triphosphate. And also remove the coenzyme A, remove coenzyme A as a result formation of succinic acid. Succinic acid. Now, if you see this third diagram, no coenzyme A. You can see here, no coenzyme A. So succinic acid formed. And seventh reaction again oxidation. Seventh reaction again oxidation. But instead of NaDH2, here the flavin adenosine diphosphate, not nicotic acid. Flavin. From the flavin adenosine, here also same hydrogen. So here the H from 1,1 hydrogens. 1,1 hydrogens here this added to the 1,1 hydrogen added to the FAD as a result formation of FADH2. As a result formation of FADH2. You can see in this here the H, no hydrogen groups are there, in only CH, only CH. But in succinase CH2, CH2, you can see here, in succinase CH2, CH2. But after remove one hydrogen, it becomes CH and CH. Rest of the common, COO minus carboxylic acid group common. Carboxylic acid group common. So seventh one, oxidation form. And again hydration process added water with help of fumarase enzyme added water as a result formation of malic acid, malate. As a result formation of malic acid, malate. So again add water, that means CH becomes CH2, and also hydrogen here the one OH also, hydroxyl group also added. So hydrogen H2O, right? So this is called as malate form. This is hydration is the eighth one. Ninth one again oxidation. What happened in again oxidation? NAD after added hydrogen to the NAD convert into NADH plus H plus. Convert into NADH plus H plus. And this process called also oxidation. And here the malate dehydrogenase. Malate enzyme name is malate, means from the malate hydrogen group removed. That's why it is called as a malate dehydrogenase. The simple remember, no need to mug up, no need to rote memorizing here. The simple, from the malate hydrogen group removed. Removed means dehydrogenase. So simple malate dehydrogenase. So after oxidation, what is formed? Oxaloacetic acid. Now we started the Krebs cycle from the 4 carbon oxaloacetic acid. Now again end with 4 carbon oxaloacetic acid. Clear? Hello?
Yes, sir.
Yes, sir.
So if you see anywhere in oxidation process, enzyme name is common. What is the enzyme name common? Dehydrogenase. From the where dehydrogenation places, for example, first oxidation. From the isocitrate to the substrate. From the isocitrate, hydrogen remove. That's why isocitrate dehydrogenase. In second, fifth reaction, from alpha ketoglutarate is the substrate, hydrogen removed. That's why it is called as alpha ketoglutarate dehydrogenase. Likewise from succinic acid, succinate dehydrogenase. Likewise from everywhere, from the substrate to hydrogen remove, means remove of hydrogen oxidation. That's why called as oxidation, dehydrogenase enzyme involved. So total 9 reactions, right? From start condensation to oxidation, total 9 steps, 9 reactions are there. My question is, how many ATPs are produced in Krebs cycle? How many ATPs are produced in Krebs cycle? See the cycle and tell me. How many ATPs are produced?
18. 18.
Huh?
180.
Direct ATP 1, right? But you don't forget, this is for one pyruvate reaction, right? Hello?
Yes, sir.
This Krebs cycle happened for one pyruvate. But you tell me how many pyruvates are produced?
2.
Glycolysis. 2
Two.
pyruvate. So another cycle.
2 pyruvic acid.
2. That's why how many ATPs here produced?
2.
Very good. 2 ATPs are produced. Let's change the color. 2 ATPs are produced. Direct ATP 2. Then how many and another type of formation of ATP through the NADH2 and FADH2, right? Already we discussed in glycolysis. First of all, my question is, how many NADH2 are produced? How many NADH plus H plus are produced in Krebs cycle?
3, sir.
3, right? 3. But you always check this 3 NADH plus H plus for one pyruvate. Then
For
right,
2, 6.
6. Very good. 6 NADH plus H plus. 6 NADH plus H plus. And how many FADH2 are formed?
1, 2 times 2.
Very good. 2 FADH2 formed. So direct ATP 2, that is clear. So indirect ATP through the NADH or FADH2. Remember one thing. In glycolysis also someone told that one NADH2 enter into the oxidation phosphorylation cycle, 3 ATPs are produced.
Yes, sir. Yes.
NADH2. For 1 NADH2 means 1 Krebs cycle NADH2, 1 NADH plus H plus
3
entered
ATPs.
the ETS cycle through phosphorylation, oxidative phosphorylation. 3 ATPs are produced.
Yes, sir.
3 ATPs are produced for 1 NADH plus H plus. Now
Oh.
tell me, through the NADH plus H plus, how many ATPs are produced?
18.
18, sir.
Very good. 18 ATPs are produced. 18 ATPs are produced. But FADH2, like glycolysis, if FADH2 enter into the electron transport system through oxidative phosphorylation,
2
2
ATPs.
ATPs are produced for 1 FADH2. 2 ATP produced. Now tell me how many ATPs are produced here?
4.
4 ATPs are produced. 4 ATPs are produced. Now tell me how many total ATPs, direct ATPs 2, right?
24.
Through NADH plus H plus 18.
24.
Through FADH2. Through FADH2 how many?
24.
4.
4 total.
24.
24 ATP in Krebs cycle. What about connecting link?
2 ATPs.
This connecting link means actually it is not clear here. After pyruvate PU, pyruvate to form as a CoA, acetyl CoA acetyl CoA NAD plus convert into NADH plus H plus. Is it clear? Is visible?
Yes, sir.
NADH plus H plus is clear. Is it visible
Yes,
or not?
Yes, sir.
Extended out NADH plus H plus extended out. Is it clear or not?
Yes, sir.
Right. NAD plus converted NADH plus H plus. So here also NADH plus H plus formed in connecting link. Likewise, how many NADH plus H plus formed here?
2
280.
2 NADH, sir.
Very good. 2 NADH2. Then, like a
Four.
Krebs cycle, this NADH2 also, if enter into the electron transport system, 3 ATPs are formed. 3 ATPs are formed. Now tell
6
me how
total.
many
6.
total ATPs produced?
6.
Very good. 6 ATPs are produced. Remember, connecting link, 6 ATPs are produced. Clear? So glycolysis, just remember, just check through the glycolysis, how many ATPs formed?
28.
6.
In glycolysis only,
24.
6 ATP. Connecting link,
6.
6 ATP. Is there any direct ATP in connecting link? Is
No,
there any
sir.
direct ATP formation? Direct
No,
ATP
sir.
means from
No,
ADP to ATP.
No,
No.
sir.
No. And how many ATP from Krebs cycle?
2 direct. 4.
2 direct. Through
4.
oxidation, through oxidation 22. Right, 18. And through FADH2 oxidation 4.
4.
Total altogether, total how many?
24.
24. For one glucose molecule, if the one glucose molecule oxidized, how many ATPs are produced? Total means from
20.
glycolysis through connecting link, Krebs cycle
36.
38. 38.
36.
36.
36 ATPs are produced. 30, but in some authorized books, it is some ATPs are changed. That means actually, as per Tej and Jaigar book, they mentioned clearly not for NADH2 oxidation, not produced 3 ATPs. They stated that for 1 ADH2, 1 NADH2, only 2.5 ATPs are produced. Only 2.5 ATPs are produced as per Tej and Jaigar book. But remember this, as per Tej and Jaigar, 2.5 ATPs for 1 ADH2. For some other books given for 1 NADH2 clearly 3 ATP. But remember the 2.5 in our mind. But in examination also you can write 3 ATPs. 3 ATPs, you can write 3 ATPs. Apart from 3 ATPs, you may mention 2.5 ATP inside bracket Tej and Jaigar. Tej and Jaigar book 2.5 ATP. 2.5 ATP. Clear? Any doubt in this?
No, sir.
Can I move oxidative phosphorylation?
Yes, sir.
Very, very important. This glycolysis or Krebs cycle or oxidative phosphorylation. Please be silent. Hello. Krebs cycle, ETS cycle, anyone may give chance to formation, give as a essay question. Hello. Please be silent. Thank you. Right. So another important topic here, oxidative phosphorylation. So for matrix, Krebs cycle takes place in matrix, but in cristae, oxidative phosphorylation takes place in the foldings, cristae place. If you see cristae clearly in this diagram, mitochondria in cristae, this is outer membrane,
Inner membrane. Yes.
outer membrane here, intermembrane space,
Is drawn up.
inner membrane,
Drawn up.
matrix, inner membrane matrix. So this is only folding parts. This is only folding parts. Outer membrane, intermembrane, and inner membrane. Outer in this diagram, this folding. This is intermembrane between the place. Outer membrane. Inner membrane. Clear? Inside the inner membrane, this is matrix. Are you clear? Hello? This is one folding diagram.
Yes, sir.
Outer membrane,
Inner membrane.
intermembrane space between the place. This is the inner membrane, and inside the inner membrane matrix. Clear?
Yes, sir.
Yes, sir.
Right. Right. So here one, let me change color. NaDH2. Already you know how NaDH plus H plus formed. NaDH plus H plus in Krebs cycle, in glycolysis, in connecting link. Clear? And also FADH2 formed here. Remember here, FADH2 and glycolysis NaDH2, listen carefully. Glycolysis NaDH plus H plus or NaDH2 start the cycle from the second complex. Before that, you should remember from oxidative phosphorylation, 5 complexes are there. Hello, please. Hello. How many complexes are formed? How many complexes are there in oxidative phosphorylation? How many
5.
complexes involved? 5. Complex 1. Complex 2. Complex 3. Complex 4. And complex 5. This one. This is complex 5. Let's change color. Complex 5. This is complex 5. Clear? So total complex is clear?
Yes,
Right.
sir.
Now first let's take Krebs and connecting link NaDH2. Krebs and connecting link NaDH2. Krebs are connecting link NaDH2. Connecting link NaDH2. If enter into these complexes factors, complex factors. After oxidized, that means NADH plus H plus oxidized into 2H plus and 2 electrons. 2H plus 2 electrons. That means NADH2 2 electrons loss. Apart from that, this 2 hydrogen also lost. This 2H plus. Now this electron, released electrons sent toward the complex 1. Right? From complex 1 to the transfer to the complex 2. Are you clear? Hello?
Yes, sir.
Yes,
So
sir.
first, after release these 2 electrons sent to the complex 1. From complex 1 to the complex 2. Remember here one thing, from complex 1 to complex 2 to transfer 1 electron, actually here from 1 by 1 electrons are transferred to the 1 to 2 complex 2. For 1 electron, remember very very important, for 1 electron transfer from the complex 1 to the complex 2, 2 protons. How many protons?
2 protons.
2 protons sent into the intermembrane space. How
Okay.
many protons? 2 protons sent to
protons.
the intermembrane space. This is for 1 electron, right?
Yes, sir.
Later, another electron also transferred into the first to second complex. So here also same, another 2 protons formed. 3 protons. Total, how many protons are formed here?
4.
4 protons formed. Clear. From second complex to the third complex, to reach the third complex also same reaction. This here involve ubiquinol cycle. In what happened in ubiquinol cycle? First the ubiquinone after received 1 electron, it received 2 protons from the matrix. 2 protons, already here the protons are formed, these protons. 2 protons from the matrix, 2 protons from the matrix to the, as a result, ubiquinol formed. Ubiquinol. And now this electrons again sent into the complex 3. Again sent into the complex 3. Here also same. From 1 electrons sent into the complex 3, 2 protons sent to the intermembrane space for 1 electron. So always you remember your pair electron, pair not 1 electron, but they are always sent 1 by 1. But after another electron through the ubiquinol cycle, another 4 protons, sorry, another 2 protons sent into the intermembrane space. Then how many protons are remained in intermembrane through ubiquinol cycle?
4.
4, right, that's it. So anyway, from the complex 3 to the electron sent in the cytochrome C. Cytochrome C is the mobile carrier. That means it is shifted to 3 to 4 from mobile carrier. This C sent the electron toward the complex 4. At the complex 4 also what happened? If the electron transferred to the complex 3 to the 4, here also same 2 protons, 1 electron, 2 protons are sent into the intermembrane space, right? Hello?
Yes,
And for another electron also, actually here the 2 protons was supposed to sent into the intermembrane space, but instead of that, to sent into the intermembrane space, these protons utilized for the water formation. These protons utilized for the water formation. So how many protons are sent into the intermembrane space through the complex 3 to 4? You remember from the complex 1 to the complex 2, 4 protons, right? Complex 2 to complex 3 also 4 protons.
Okay.
Actually, complex 3 to complex 4 also, how many protons need to send? 4. But instead of 4, how many protons are sent into the intermembrane space here?
3, sir.
You see here, how it is 3?
2, sir. 2, sir.
Only 2. So
Only
what
2
about
protons.
remaining 2 protons?
Water formation.
Right, remaining 2 protons utilized for the water formation
Water
here.
molecule.
Water formation.
Okay.
Electrons, all that means the cytochrome C to the electrons, oxygen received, oxygen to the protons, as a result formation of water molecules formed. Now tell me how many ATPs are produced? Sir, how many protons are produced in intermembrane space total?
8,
From
sir.
complex 1 to 2, 4, right here 4, ubiquinol 4, 4
8.
plus 4,
8 and 8 plus 10.
plus 2,
10.
total
10, sir.
10
10, sir.
protons. Right. 10 protons are produced in intermembrane space. Due to the proton gradient difference to the matrix and intermembrane space, these 10 protons always move towards the natural place of matrix, mitochondrial matrix, but it is this layer, inner membrane, acts as impermeable layer. That's why it choose to another where complex 5, that is called coupling factor F0 F1. F0 F1 complex. F0 F1 complex. The function of F0 F1 complex for 3 protons. For 3 protons travel through this complex, the head region, this head is head region. This head region rotate as a result formation of 1 ATP. 1 ATP for 3 protons. 3 protons travel through these complex protons. How many ATP are produced?
180.
1 ATP formed. Right? Right?
Yes,
So
sir.
here the
Yes,
now
sir.
tell me from 10 protons how many ATPs produced? For 3 protons 1
10
ATP.
ATP. Okay.
For 10 protons
3 and
3
3, sir.
leave it 1 protons total 3
3.
ATPs 3 ATPs are produced. 3 ATPs produced. As we discussed earlier, Krebs cycle. If Krebs cycle NADH plus H plus enter into the electron transport oxidative phosphorylation, how many ATPs are produced? 3 ATPs produced. How the 3 ATPs are produced? Likewise, at the 4 protons, 2 protons, total 10 protons are formed. These 10 protons, if travel through this coupling factor, 3 ATP formed. Is it clear?
Yes. Yes, sir.
Then what about glycolysis? Glycolysis and FADH2. FADH2. How it is formed? FADH2. So you can see here. You can see here. Already told you FADH2 and glycolysis NADH2. FADH2 and glycolysis NADH2. This both start from complex 2 only. Not from complex 2 only. But not complex 1. That's why you can skip 4 protons. Right? Now tell me how many protons are formed. You tell me if the FADH2 or glycolysis NADH2 enter into this ETS cycle, how many protons are produced in intermembrane space?
8.
8.
How it is 8?
10.
That means this portion is skip. This portion. Complex 1, 2, 2 is skip. Direct from the 2 to 3. From complex 2 to complex 3 ubiquinol, how many protons are sent into the intermembrane space?
4 protons.
Right, that's it. And from the complex 3 to complex 4, how many protons are sent into the intermembrane space?
4.
4.
Actually was to send 4, but instead of 4, how many protons are sent into the intermembrane space?
2.
2
Two
protons.
only. What about remaining 2?
Water formation.
Formation
Right,
of
water
water molecule.
formation. So total FADH2 and glycol NADH2, total 6 protons are formed, right?
Yes, sir.
These 6 protons, if travel through this coupling factor, how many ATPs are formed?
2 ATPs. 2 ATPs.
How many ATPs
2
formed?
2, sir.
ATP. 2
Oh, very
ATP.
good. 2 ATPs are formed. That is the difference between glycolysis NADH2, Krebs NADH2. Glycolysis and Krebs NADH2. Is it clear?
Yes, sir.
Any doubt in this?
No.
Right. Thank you all.
Thank you, sir. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.